Assalamu alaikum, dear viewers. This is my first presentation of Principles of Chemistry 1. Let me introduce myself. This is Shugupta Zulkar from BS Mathematics, and I have to submit this presentation to my most respected professor, Dr. Hamad Najid. And the topic of this presentation is new COVID variants, COVID-19. The major outlooks of this presentation are COVID-19, its history, symptoms, and how it is transmitted from one person to another. Then we see what is variants, what are classes of variants, what are important variants, what are their characteristics, and then we see composition of vaccine, its side effects, how we can overcome these effects, what are vaccine challenges, and what are the hurdles, and then we see conclusion of our this presentation. So let me tell you what's coronavirus. Coronavirus disease is an infectious disease caused by SARS-CoV-2 virus. SARS-CoV-2 virus, severe acute respiratory syndrome coronavirus, is a strain of a coronavirus that causes COVID-19. The respiratory illness responsible for ongoing COVID-19 pandemic. The virus previously had a provisional name 2019 coronavirus and has also called the human coronavirus 2019. History. In December 2019, an outbreak of pneumonia of unknown origin was reported in Wuhan, Hubei province, China. Pneumonia causes were epidemically linked to Hunan seafood wholesale markets. Inoculation of respiratory samples into a human airway epithelial cells, Viru E6 and Hu7 cell lines, led to the isolation of a novel respiratory virus, whose genome analysis showed it to be a novel coronavirus related to SARS-CoV. The global spread of SARS-CoV-2 and the thousands of deaths caused by coronavirus led the World Health Organization to declare a pandemic on 12 March 2020. So these are some major symptoms of coronavirus. Fever, 81.2%, cough, 58.5%, fatigue, 38.5%, dyspnea, 26.1%, sputum, 25.8%. Transmission. Frequent sneezing and dry coughing exhibited by COVID-19 patients generate viral plumes of thousands of droplets per cubic centimeter. Since SARS-CoV-2 infection is believed to be transmitted by aerosols and or droplets, it is imperative to assess their particle characteristics, aerodynamic behavior, and their propensity to bypass various physiological barriers to enter the host body. Now we see what are new COVID variants. First of all, we need to know what is variants. Genomes that differ from one another in genetic sequence are called variants. Variants can differ from each other by one or more mutations. When a phenotypic difference is demonstrated among the variants, they are called strains. So now we see classes of variants. First of all is a variant of interest. This class includes variants with specific markers that are associated with changes to receptors binding, decreased antibody neutralization developed against previous infection or vaccination, decreased efficacy of a treatment's potential diagnostic impact, or predict increase in transmissibility of decreased severity. This class also requires increased sequence surveillance, increased laboratory characterization, and epidemiological analysis to check the disease transmissibility and severity, the risk of reinfection, and protection against vaccination. Here you can see the structure of S protein. And the next is a variant of concern. This class includes variants which demonstrated high disease transmissibility, more disease severity, including hospitalizations and deaths, remarkable disease in antibody neutralization, decreased effectiveness of the treatments, and diagnostic detections failure. This class also requires increased effects to control spread by developing testing kits, increased research to ascertain the vaccine and the treatment's efficacy against variants. The third variant is a variant of high consequence. This class includes variants which have evidence that the prevention and medical measures have a significantly decreased effectiveness compared to previously circulating variants. There are no variants in this class. So now we see new COVID variants. First of all, we see UK variants. B117, also known as VOC 2020-12/1, was detected in September 2020 in the UK. It has 23 mutations compared to the original strain found in the Wuhan, China. 8 of these mutations were found to be in the S protein. Its notable mutations were N501Y, 69 with 70 deletion, and P6818. The 9501Y mutation appears to allow for the S protein to bind more tightly to the ACE2 receptor. The next variant is South African variant. The B1351 variant, also known as 501YB2, was first identified as early as October 2020 in Nelson Mandela Bay in South Africa. By December 2020, it was also detected in Zambia. By April 1, 2020, there were 323 reported cases in 31 jurisdictions in the USA. There are 23 mutations with 17 amino acid changes, but the noticeable mutation in this variant are K417N, E4847K, and 9501Y on the S protein. It is suggested to have increased transmissibility and the most commonly seen in young people without underlying disease. The next variant is a Brazilian variant. P1 variant, also known as B11281, was first detected in North Brazil in the city of Manaus in the Amazonas state in the December 2020. By the January 2021, it was identified just outside Tokyo at the Haneda airport among 4 travelers from the Brazil during routine screening of passengers in the samples analyzed at the National Institute of Infectious Disease in Japan. The next variant is a Brazilian variant. The Brazilian variant B1128 was initially found in the state of Rio de Janeiro in Brazil and in the P2 lineage. Unlike P1 variant, B1128 variant has only one notable E484K mutation in the S protein. The therapeutic and vaccine efficacy has not been reported yet. Similar to B1351 and P1 variants, the vaccine might be ineffective against this variant. The next variant is US Midwest variant. It was detected in Ohio, followed by other Midwest states in December 2020 and January 2021. This variant has mutations on the S protein, M proteins, and on the N proteins. Another variant with the mutation SN501Y, a marker of the B117, with no other associated mutations with that strain has been identified. Currently, there is not an evidence of increased transmissibility of this variant. The next variant is US San Francisco Bay Area variant. These variants was first identified in California in February 2021. Notable mutations in a B1427 are L452R and D614G, while mutations in a B1429 and S131, W152C, L452R, and D614G Both variants have a 20% increased risk of transmissibility and reductions in a therapeutic efficacy. It was identified last year across the USA and Europe. It is a variant with a mutation in aspartame. In January 2021, it rose rapidly as a cause of cases across several countries in California. The next variant is US Southern California variant. It was first seen in July 2020 in Southern California and detected amongst population samples of the same region in October 2020. Its notable mutations are OR51A, I4205V, ORF1B, D1183YS, S131, W152C, and L452R. The binding of the aspartame could be made easier by the later three mutations. The next variant is B1526 and B1525. These variants was first identified in New York, USA. The notable mutations are E484K, S477N, while E484K decreased antibody response. S477N increases attachment process. The next is a double mutant variant. This variant is first detected in India. As two mutations are seen in the same virus, this variant is called a double mutant virus variant. There was a significant increase in COVID-19 cases in India. The first case in the USA was identified in San Francisco in April 5, 2021. The notable mutations are E484Q and L452R. These variants are at increased risk of transmission and also resistance to vaccination. The next variant is a triple mutant variant. In addition to E484Q and L425R, in double mutant variants, the new triple variant discovered in April 2021 is characterized by the deletion of two amino acids H146DEL and Y145DEL in the S protein. As of April 21, 2021, a total of 1189 samples were tested positive in Maharashtra, Delhi, West Bengal, and Chhattisgarh, India. Similar to the other variants, triple mutant variants have high transmissibility. The next variant is 20A-EU1 or SA222V. The 20A-EU1 variant has non-terminant domain mutations which do not play a direct role in receptor binding or a membrane fusion. This variant was initially identified on 20 June 2020 in Spain, but rapidly spread across Europe and many countries. The next is the 20A-EU2. The 20A-EU2 variant was found in France in June 2020 and has become the second dominant variant in Europe. The notable mutations are S477N, E484K, and N501Y, which demonstrated slight increase in ACE2 binding resistance to multiple antibodies and a covalent serum. The conformodis increased in effectivity as measured by soluble MACE2. The next variant is 20A or S439K. The 20A S439K variant was initially found in Ireland. This variant has SN439K mutation with the deletions of amino acids at the position 69 and 70 of the S proteins that result in an increase in ACE2 binding, resistance to antibodies, and the covalent plasma. The next variant is 20A-S98F. The 20A-S98F variant has S98F mutation, which was found predominantly in Belgium and the Netherlands. The next variant is 20CS80Y. The 20CS80Y variant had 18 nucleotide mutations, possibly related to the apolipoprotein B editing complex APOBEC, like editing within the host, which is found in at least 10 countries in Europe. The next variant is 20B S626S. The 20B-S62S variant has S626S mutation. This variant is found in 15 countries of Europe. This is predominantly seen in Norway, Denmark, and the UK. The next variant is 20B-S1122L. The 20B-S1122L variant has SV1122L mutation and is found predominantly in Sweden, Norway, and Denmark. The next variant is N440K. According to the latest report, another new variant, N440K, with a mutation in asparagine was emerged, which resulted in the sudden increase in case of Andhra Pradesh, India. The Center for Cellular and Molecular Biology found that this variant has enhanced binding to the ACE2 receptors 10 to 1,000 folds more transmissible and resistance to class 3 monoclonal antibodies C135 and REGN 10987. Now we see what are COVID-19 vaccine versus the variants. The Food and Drug Administration has approved 3 vaccinations for emergency use in USA, and data suggested that the mRNA vaccines responsibly provide protection against B117 COVID-19 variants. The mRNA vaccines, Pfizer and Moderna, were authorized in USA before the identification of South African variants strain in the country. The next topic is structure and composition of vaccine. So the major points are antigens, adjuvant, preservatives, stabilizers, surfactants, residuals, and diluent. So first of all, we discuss about antigens. This is the active component that all vaccines contain, created to resemble or mimic a particular disease or infection, or a pathogen or antigen's purpose is to provoke your immune system. But doing that, it's laying the groundwork for the future immune responses to an actual infection by the pathogen, in this case, the COVID-19 virus. Preservatives. If a vaccine comes in a single dose, it may not contain any preservatives, but some are delivered in large vials designed for multiple shoots, in which case they need something to prevent the vaccine spoiling once exposed to air. One of the most commonly used preservative is 2-phenoxyethanol, which is used as a perfume fixative, insect repellent, and an antiseptic. And here you can see the diagram of a phenoxyethanol. Stabilizers. Sugars, amino acids, gelatins, and proteins are all used as stabilizers in vaccines. They are there to prevent chemical reactions taking place within the vaccine. One of the often used proteins in a recombinant human albumin. This is made from yeast, and although it resembles human serum, it contains no human or other animal material. And there is a diagram of amino acids. You can see it. And next, there is the structure of lactose, glucose, its chemical structure and its physical structure. You can see it clearly. And next, you can see structure of proteins. And this is primary structure, and in the mid, there is secondary structure, and there is tertiary structure, and there is a quaternary structure. So the next is surfactants. Once stabilizers stop vaccine components mixing and reacting, surfactants are there to keep all the ingredients mixed together. Without surfactants, there could be a separation of ingredients leading to the sediment or clumps forming. So, the next is a different manufacturing process use different non-active ingredients. Trace elements of these non-active elements are present in the final vaccine and are known as residuals. There may be things like yeast or egg protein or even antibiotics. There are in such tiny amounts that are made in a parts per billion. And there is a structure of yeast cell and also the structure of an egg cell. You can see it clearly. And next you can see a tetracycline, which is an antibiotic drug, its molecular structure. And the next is the diluent. The greatest ingredient by volume in a vaccine is the diluent. As the name suggests, it is a liquid used to dilute the vaccine to achieve the right concentration. Usually, the vaccine diluent is a sterile water. Adjuvants. Some vaccines contain small amounts of aluminum salts to help trigger and stronger immune responses. These adjuvants work by stimulating immune cells near the injection site. Aluminum is sometimes the subject of the alarmist concerns, but people routinely come into the contact with aluminum through food and drink, and it has been shown not to cause long-term health problems, the WHO says. Here you can see the structure of aluminum. So next we see what are the major side effects. The most commonly reported side effects were fever, headache, fatigues, pain at the injection site. Helpful tips to relieve side effects. To relieve pain or swelling in the arm where you got the shoot, apply a clean, cool, wet washcloth, cover the area. Use to keep moving your arm. Also, if possible, get some rest. To reduce discomfort from fever, drink plenty of fluids, dress in a comfortable clothes. Talk to your doctor about taking over the counter medicines such as ibuprofen, acetaminophen, aspirin, or and antihistamines. Ask your child's healthcare provider for evidence advice on using a non-aspirin painkiller. Next, we see vaccine challenges. More than 75 billion people have been fully vaccinated as of April 13, 2021. Since December 14, 2020, during the same duration, there have been 5,814 vaccine breakthrough cases reported across the USA. 45% of the total cases were female, 29% were asymptomatic, 45% were over 60 years of age, 7% of the total cases were hospitalized, and 1% died. And these are major hurdles. Pregnant and breastfeeding women, immunocompromised, diverse race, and ethnicities have been underrepresented populations in vaccine studies. Vaccine refusal have been an issue physicians have had to deal with even in a pre-pandemic times. So, conclusions. The emergency variants not only result in increased transmissibility, morbidity and mortality, but also have the ability to evade detection by the diagnostic test, exhibit decreased susceptibility or treatment, including antivirals, monoclonal antibodies, are Convalescent plasma possess the ability to cause reinfections in a previously recovered and vaccinated individuals. 40.7% of the total populations in the USA were fully vaccinated. The USA ranks 8th in terms of percentage of the population vaccinated. Israel ranks first with 57% of the total population vaccinated, followed by Bahrain in Arabia. And here is a reference of websites from which I have taken the whole material. Thank you very much for your special attention.